Hey guys. Alright. Can everybody hear me? Okay, good. Okay, so today we are broadcasting live from my lab here on the University of Missouri campus. For those of you who may be watching this later, my name is Dan Leuchtman. I'm a PhD student in the Leuchtman and Gassman labs here on campus. And we are broadcasting live with Miss Cooper's 5th grade elementary class at Mill Creek Elementary. So, today we're going to be talking about my work for a little bit, and so, like I said, I work for Dr. Leuchtman and Dr. Gassman, and Dr. Leuchtman and Dr. Gassman are both molecular biologists that study plants. And so they may sound kind of complicated and scary, but all it really means is that we study specific molecules and what they do for the plant. And so, as you guys learned before, we know that plants need light, they need nutrients, they need water, and they need a place to live, so they need soil. And so, light's a big thing that Dr. Leuchtman studies, and so plants need to find the best light around, and believe it or not, they can actually behave and try and find it out. So here's a video. Look at it for a second. Does it start automatically? So here you guys see, and you may have seen this image before, you see some maize or corn seedlings that are placed in a pot. And in the middle they put this light bulb, and so when the room is in complete darkness except for that light is on in the middle, all the plants will grow towards that light. And as soon as the normal lights come back on, so the ceiling lights, they start to grow towards those, and we call this the phototropic response, or we call the process phototropism, so how plants forage and find out light. And so while this may be kind of a silly example, it's actually kind of fun to watch. This actually gets pretty important in nature. So I want you guys to think about So I want you guys to think about where this may be important in nature. So you guys know that plants grow around each other, and so if you're a big tree, you may not have to worry about this so much because you're closer to the sun than most other plants. But if you're a seedling just sprouting up, this becomes very important. So here's an image of a dense forest canopy, and you can see big tall trees and vines and stuff growing all over the ground. But as you can see that there's those tiny little rays of light poking through. And if you're a seedling that's growing kind of towards the left of the screen where there's not a whole lot of light, they're going to try and grow towards that beam that's coming through there in the middle. And so that's where this process becomes very important in nature. Cool. So next we're going to talk about what Dr. Gassman studies for a little bit. And so as we mentioned before, plants need soil to live in, and plants aren't the only thing that live in the soil. So there's tons of other creatures and animals and microbes that live there too, as you guys have been learning about this year. So there's bacteria, there's fungi, there's viruses, there's insects, there's other animals, there's a whole lot of stuff. And so here is a microscopic image, so you guys can see down here that little 3 and that funny looking symbol next to an M, so that is 3 micrometers. So there are 1,000 micrometers in 1 meter, so this is very, very small. And as you can see on the surface of this plant leaf, there's couple cells there, and those teeny tiny little white dots are bacteria, and they're trying to get into the plant. Most plants can actually defend against this bacteria, so they do just fine, but that is mainly because they prevent anything from getting inside the plant. But sometimes, you can see the little opening right there, sometimes bacteria and other organisms find a way to get into the plant. But again, plants can even detect that and kind of sound the alarm to the rest of the plant to tell it to, hey, someone's trying to get in, we should do something about it. But it's when these microbes and other organisms disrupt that actually causes disease. If you guys see a really sick tree outside, it's because it was unable to defend against those specific microbes. And all this happens within the cell, and I want you guys to kind of picture that for a second because you're seeing a picture of that one right now. And so everything that we're going to be studying as microbiologists for the day is within the cell. And so the first thing we're going to do is give you guys some basic cell biology. So we're going to go over to the whiteboard and do some basic stuff real quick. Camera then. So I want you guys to keep in mind that cells are three-dimensional images, so they're kind of like a little bubble. So what I'm going to be drawing on the board is two-dimensional, but keep in mind that real cells are three-dimensional. Okay. So the first part of our basic cell is called the plasma membrane. If you guys are following along on your worksheets that we handed out, that may be important. Next what we have are organelles, and these are small little parts of the cell that do things for the cell as a whole. So think of the word organelle, so it's kind of like organs in our body. So just like we have a heart, it does a specific job for our body, so our heart's job is to pump blood throughout the rest of our body. Organelles are little parts of the cell that can do different jobs for it. So like I was saying, it can help store energy, help create energy, other things like that. Just like we have organs that do specific jobs, like our heart pumps blood and our liver helps purify that blood, kidneys, so on and so forth. So that's what organelles do. So next we have the nucleus. And that's where DNA is stored, and we'll talk about that in a little bit. And lastly, we have the cytoplasm, and that's this in-between stuff and everything that's within. Yeah. So all that good stuff. Okay, so next I want to talk about for a second about what these cells are made of, because that's important too. So just like these different structures are important for the different things they do, they're made of different molecules and those can do different things. So one of the first things cells are made of are carbohydrates, and you guys may have heard that word before. So one common one that I know that you're very fond of is sugar. So sugar is a carbohydrate. And these are chains of molecules that the cell can use to build different things and help do different things within the cell. And those are all over the place within the cell. So there's some in the nucleus, some in the plasma membrane, some within the cytoplasm, so on and so forth. There's also lipids, and lipids, what you guys would think of as fats. So if you were to dump some oil in a pot of water at home, they would form little bubbles, and those little bubbles are kind of like what cells are. So the main place within the cell that you would find those are on the plasma membrane, so right along here. And they're also within the organelles and some places within the nucleus, and so on and so forth. So next we're going to talk about is proteins. And I know you guys have heard that word before. You'll probably think of an example as a nice juicy steak has a lot of protein in it, and that's very true. But proteins are small molecules that can do things for the cell. So they're kind of like little machines. And we'll talk a little bit about that once we start doing some stuff at the bench later. The last thing we have is nucleic acids. And this is actually what DNA is made out of. And so that Na in DNA is actually what stands for nucleic acid. And that's located within the nucleus. There's some other nucleic acids that are within the rest of the cell too. But so now you guys have the 4 basic components of your basic cell and 4 things that the cell is made out of. And so we've gone through quite a lot, so I'm going to give you guys some question and answer time really quick to make sure that we're all up to speed. So you guys talked a little bit about DNA, and DNA is a very interesting molecule. So it is composed of chains of nucleic acids. Here, I'm going to take this off for a second. So it's composed of chains of nucleic acids that the cell uses as information. So you guys read a book. You have different letters. The different letters make different words. The different words make pages, paragraphs, so on and so forth. So you can kind of think of DNA as like the letters almost. And so what you're looking at right now is a picture of human DNA. So DNA exists on these little structures called chromosomes, and so this image was made when someone took DNA out of a cell, they stained it and purified it, and kind of manipulated it in a way so that we could actually see it and look at it. And so each one of those little worm-like things is what we would call a chromosome, and it is a collection of DNA. And so in human DNA we have 23 different kinds of chromosomes, and so we have a total of 46 within each and every one of our cells. And so that codes for about 20,000 genes, which is a pretty decent amount. But we're not exactly sure how this stuff works, and that's part of one of the big things that biology is trying to solve, is exactly what is DNA or other nucleic acids and how they work, and how you go from these tiny little worm-like things to whole organisms. And this can be very complicated. So for instance, this is an image of an Arabidopsis genome, and so I forgot to mention before, so a genome is all of the chromosomes of an organism put together. So here you have the human genome, so all of the chromosomes that would be within one of our cells, and then here you have the Arabidopsis genome. So as you can see, so Arabidopsis only has 5 different kinds of chromosomes, or we have 23, so that's a lot less DNA, but Arabidopsis genome also codes for about 20,000 different genes, and so that is one of the big problems that we're investigating as scientists, is how you go from an organism such as myself to Arabidopsis. And so you guys have seen this little guy before, and so for those of you watching who may not know, Arabidopsis is a small model organism, a plant model organism, and so a model organism is just one that is small, easy to work with, there's lots of tools available for it. It's cheap to use, and so there's lots of different models. So mice and rats are models for human biology, and so Arabidopsis is one of the models for plant biology. And so I think you guys know that, but I was just adding that just in case. And so that's one of the big issues that we're working out, is so how 20,000 genes can go from a small little plant to myself here having a conversation with you guys. And so that's part of what we study in the Leuchtman and the Gassman labs, is how DNA, which like I said is information, tells the cell what proteins or what molecules to make, and most importantly when to make them. So each and every one of your cells has the DNA that tells you how to make an eyeball, but you wouldn't want to make an eyeball in your toenail, would you? So when you make those proteins and molecules is just as important as having it and knowing what to make to begin with. And so that's where I'm going to need your guys' help today. So we're going to study, I'm studying a segment of DNA that codes for a protein, and I'm not exactly sure what this protein does. And so when you're studying just a single molecule like a protein, you kind of have to think of a new approach on how to do it. So if you guys wanted to study something within our cell, you would just kind of go up and look at it, wouldn't you? But it's not that simple. So imagine you want to study just the red balloons in your bowl of Lucky Charms. Only a cell is more than just a couple handfuls worth of protein. It's actually kind of more, you can think of it as a swimming pool full of these different molecules. So imagine you're trying to study just the red balloons in a swimming pool full of molecules, but unfortunately you can't just dive right in and go looking around for them. So you're trying to study just the red balloons in your Lucky Charms in a swimming pool full of Lucky Charms, and you're only allowed to look at it from a helicopter. So this kind of gets complicated, and you need to find a way to identify your protein. So if there is some way that we could take our protein and make it do something unusual, so that we can find it in the cell and see where it is, because that's one of the important parts, one of the first steps in finding out what your protein does is finding out where it is. Where'd that black light come from? Okay, so you guys may not actually be able to see it, and human eyes can't detect it, but these black lights emit UV radiation. But sometimes when that UV radiation hits the right molecule, so for instance the molecules that are on my lab coat, they reflect in a color that we can see. It kind of looks like it glows. It's kind of interesting, isn't it? So maybe if we somehow attach one of these molecules that glows under UV light to our protein, we could detect it. And that's exactly what we're going to do today. So first thing we need to do is we need to get some of the DNA for our protein, and then we can mess with it a little bit. So we're going to go over to my lab bench real quick, and I'll show you guys how this works. Alright, so welcome to my lab bench. This is where I do most of my work. And so like I said, we need to take the DNA that tells the cell how to make our protein, and we need to kind of manipulate it a little bit. So the first thing we're going to do is we need some of that template DNA. So we need to make a bunch of copies of it. So we need to get what we need to copy first, right? So we do that by kind of taking our plant, and we are going to grind it up. So here you have a small Arabidopsis plant, and you guys have seen this before, and I'm going to pulverize it. So we do that in this little device called a mortar and pestle, and these have been around for a long, long time. So I'm just going to put that in there, and then I'm going to dump some liquid nitrogen in there. So liquid nitrogen is extremely cold. It's liquid at room temperature. See, it's kind of, well, I'm sorry, it boils at room temperature. So like I said, this is very, very cold, and so this looks like a boiling pot of water, but that's only because nitrogen boils at room temperature. And so this is within this container right here, it is about negative 110 degrees, so it's pretty darn cold. So it is going to freeze that plant. And after all this liquid goes out, we're going to grind it up. We don't need all that. Now we have a frozen plant, and so now I'm going to grind it up. It's crunching just like a potato chip, pretty much. You see that? Come on. Need to wait for all the nitrogen to boil out really quick. Close. There we go. Alright, now we grind it up. What you end up getting is this powder. And so within that powder is everything within the cell. So like we said before, there's carbohydrates, lipids, proteins, nucleic acids, and we want to just get the nucleic acids out. So we're going to have to extract that somehow. So how we do that is take one of our handy dandy tubes. This is where a lot of biology happens, within these tiny little plastic tubes, and we're going to get some of this plant powder in here. Okay, not bad. Maybe do a little more. Cool. Okay. Next we need to add some liquid that's going to help us separate everything else except for the nucleic acids, because again, that's where the DNA is and that's what we're interested in. So I'm going to add some of this stuff right here, CTAB buffer, and to do that I'm going to use my handy dandy pipette, if I can find it. I'll use this one. Okay, so this is a pipette. These little numbers on the side tell me how much liquid, so this is in microliters, so there are 1,000 microliters in 1 milliliter of water, or 1 microliter is 1/1,000,000th of a liter, so extremely tiny amounts of liquid. So we're going to use this. I'm going to use about 200 microliters, and I'm going to take it from our CTAB buffer. I'm going to mix it in. Okay, and we mix it up. And you get this little tube of green goopy stuff, which is basically just pulverized plant. So then we need to separate this out from everything, and how we do that is we spin this around really, really, really fast in a machine called a centrifuge, and so that basically separates out all the heavy stuff to the bottom of the tube, and all the lighter stuff, including DNA, kind of settle to the top. And so I've done that earlier today, and I'm going to go grab it real quick. So, like I said, all the heavier stuff falls to the bottom, so all the green stuff is a lot heavier, and then the clear stuff at the top is where the DNA is. Okay. So I'm not interested in all the stuff that's down here. I'm only interested in the stuff on top, so I need to extract that somehow. And again, I'm going to use one of my handy dandy pipettes. I'm going to slowly extract that out. Perfect. Okay. Okay. So if I just extracted the DNA out of all that plant material, what's left over in the tube? I think that's one of the questions on your guys' worksheets. Cool. Okay. So this still has some other stuff in there, and so it needs to go through a couple purification steps, and conveniently I did that earlier today also. And so this is what you end up with. There's this tiny little speck of liquid that has all the DNA that was in that plant tissue or within the cells that we just ground up in that mortar and pestle in the liquid nitrogen. So, again, the Arabidopsis genome has about DNA that codes for about 20,000 different genes on top of DNA that does a whole lot of other things. But again, we're only interested in the tiny bit of DNA that tells the cell how to make my protein. And so we need to make copies of just that tiny little piece of DNA. And so we have our template, what we're going to start with, and next we want to make a bunch of copies of that. So kind of picture a copy machine. You have a piece of paper that you want to make a bunch of copies of it. So you need a couple things. You need extra paper in order to make that copy, and then you need a printer, something like that, or a copy machine. So we have small molecules that kind of do just that. So first thing we need to add is our template. So again, we just extracted this DNA out of this plant tissue, and we're going to put a little bit in the cell. 5 microliters should be more than enough. 1, 2. Alrighty. Put that down. Extremely tiny, tiny, tiny amounts of liquid. Next, like I said, we need to add, so if we're thinking of our copy machine example, we need to add stuff to make copies with. So nucleic acids in the case, that's kind of like adding paper to a copy machine. So there's some of that in here. On top of a little protein, remember earlier I said that proteins are like little machines. So this has a protein that can both read and make new DNA at the same time. And so it makes exact copies of what you put in there. And I need to add some of that. Nice green color. Cool. So the last thing we need to add is something to help that protein find just our piece of DNA, so just our gene. And so picture you told someone you want to make a copy of just one page of a book, but you just throw a book in there, throw a copy machine in there, and throw some paper in there. Doesn't quite help. You need to at least tell them the page number, tell them something that's going to allow them to find it. And so we're going to add what we call primers there. And so there's little pieces of DNA that help that protein find the piece of DNA that we want to copy. So we're going to add that in too. Some of that. And some of that. Cool. So now we're all ready to go to make a bunch of copies. So we put this mixture in what we call a thermocycler. And so this is a machine that changes different temperatures so that it tells that protein inside, or if we're using our analogy again, our copy machine, when to turn on, when to turn off, and basically tells it when to make all our different copies. And so this is one that I did earlier today. So hopefully if our reaction worked, we're going to have a bunch of copies of just our DNA that tells us how to make that one protein. So the one gene out of the 20,000 that's in there. So I'll put this in for later. Okay. So now we have a lot of copies of our gene, so just our piece of DNA. But sometimes the protein that's in there will start and stop and kind of make little chunks of DNA, so like little pieces of the page that we want to copy, but it might not be perfect. So what we're going to do is we're going to purify this a little bit, so we only have our nice perfect chunk of DNA that we know codes for our protein. And we do that by running it out on a gel. And so, DNA is negatively charged, and you guys may have a question about this on your worksheet. So, the gel is basically just a bunch of teeny tiny little molecules that the DNA has to weave in and out of in order to go through. So if we, like I said, if DNA is negatively charged and we can put a charge through that gel, we can end up dragging the DNA through it. And so I want you to think about that for a second and answer a question. So if DNA is negatively charged, which direction is it going to follow on the charge? And are bigger pieces of DNA going to go through faster, or are smaller pieces of DNA going to go through faster? So I'm going to give you guys a quick second to answer that, and I'm going to take care of my gel real quick. This will be important in 1 second. Cool. So I'll bring this out here so we can see it a bit better. Okay. So hopefully you guys answered that question. And hopefully you found out that DNA is negatively charged, so it's going to follow the positive electrode. So if we put our DNA up here on this gel, and put the positive charge down here and a negative one up here, our DNA will try and move through this gel and it'll go towards this positive electrode down here. And hopefully you also answered that the smaller pieces of DNA are going to move through much faster, because like I said, this is just a bunch of molecules that make it difficult for DNA to kind of move through. So if we run the DNA through this gel based on a charge, it will separate it out according to size. And if we know about how big our piece of DNA is going to be, we should know where to find it within this gel. But first we have to load it in there. So again, this is a PCR I did, or this is a reaction I did from this machine earlier today. I'm going to load it in this gel. Need more than that. Okay, you guys will be able to see this. There we go. There's one lane right there. New tip. Next one right down here. Cool. Okay, next thing we need to add is some kind of reference. So there might be a whole bunch of different bands or little pieces of DNA on here, but we don't know how big those pieces are. So what we do is we add something called a ladder, which has little pieces of DNA that are, you know, there's a small one, there's a slightly bigger one, a next size, and we know exactly how big those sizes are. So we can compare our bands that we see on this gel with our ladder. So I'm going to add that again because we need to have a reference. Cool. Alrighty. So now I'm going to put this on. I'm going to turn the machine on. And now it's running a current again through our gel. So it's going to separate out the pieces of DNA based on size. And because DNA is again negatively charged, it's going to go towards this positive end and it's going to separate out as it goes. But this takes a few hours, so luckily I have a piece right here. Hi. That's random. Okay, anyways. So, this is a piece of DNA that we ran out earlier today, or a gel that we ran out earlier today. Hold on one second, guys. Sorry about that. Okay, so this is a gel I ran earlier today. So again, I put the DNA up here. I have my ladder on this side, and you can't see the DNA in here. But you can see these little dye markers. And so what I need to do is I need to cut out just our piece of DNA that we're interested in. And so I can't exactly show you an image of this, but there's a way that we can put this in a machine mix a molecule in here that will bind onto DNA, and just like our glowing molecule we had earlier, it will glow as soon as we see it. So I'm going to show you guys a picture of what this gel image would look like if we could see a picture of it. So I'm going to go over to the computer real quick and show you that. Okay. So this is a picture of our gel image, and you guys should have one of these on your sheet. And I think the question asked you if our DNA segment is about 750 base pairs, and so that's a unit of length that we use to measure DNA, which one of these bands should we cut out? Okay, can you guys hear me? Okay, so if you guys, so it's the second to last one from the bottom. Okay, that's correct. Good job, guys. Okay, so now I want to cut out just that little piece of DNA. And so again, because we put a molecule in here that will bind to DNA and then glows when it's exposed to UV light, we could use UV light to see those different bands and know which one to cut out. And I don't think we're going to be able to see it very well in here, but so I'm going to put this on what we call a UV box, which is right here. And I'm going to turn it on. Got to put this guy on because UV light is bad. Wear sunscreen. Okay. So now, what's happening is there's UV light being shown from the bottom of this box, and so that is reflecting off the DNA. So if it was dark enough in here, you guys might be able to see through the camera, but I highly doubt you're going to be able to. So I'm going to cut out just that tiny little 750 base pair band that you guys saw on the video screen. And I'll show you an image of what this would look like in the dark here in 1 second. First, I need to cut this out. So this teeny tiny little piece of gel has the segment of DNA that tells the cell how to make our protein. So this is what we're interested in. But first we need to purify it a little bit. But real quick, I'm going to show you guys an image of what this would look like to what it looked like here for me. Cool. So this is an image of what DNA looks like when it's on a UV light like that in the dark. And so you can see the little bands on there, and they're glowing that really cool kind of bright fluorescent orange color. And so when I was cutting out that gel, that's kind of what it looked like, only it looks a lot brighter when you're there in the dark. Excellent. Okay. So we need to get rid of the gel itself and all the other nonsense in here, and so we can purify this a little bit more, and I conveniently did that a little bit earlier today. And so it all comes down to this teeny tiny little speck of liquid. And so this is a purified piece of DNA. It's only our DNA that we're interested in that tells us, or tells the cell how to make our protein. And so we can do a lot of different things with this. We can chop it up into different pieces, we can add stuff to it, we can delete stuff from it. This is kind of where people manipulate DNA, is in these teeny tiny little test tubes and these teeny tiny little specks of water. And so what we're going to do is we're going to add the DNA that will tell the cell to add one of those glowing molecules, like the kind that are in my lab coat, to our protein. So that way, when we shine a UV light on our cell, we'll be able to see it within the cell. So, I've also conveniently already done that. Where did it go? This guy. Right here. Okay. So this has everything we want on it. It has the DNA that tells the cell how to make our protein and how to put one of those glowing molecules onto our protein. So now all we need to do is get it back in the cell. And so you can't exactly just take some tweezers and stick it back in there. It doesn't quite work like that. So we need to figure out a way to do this. And there's a convenient way that biologists have figured out how to do this in plants. We're going to use bacteria. So, wait a second, Mr. Leuchtman, I thought you said bacteria was bad. We talked about that a little earlier today, and hopefully you guys know that that's not necessarily true. Some bacteria is good and can actually help the plant. But this particular bacteria is actually, you may have seen it outside, and I'll show you a quick picture of that. So this is what we call crown gall, and so it kind of looks like there's this tumor kind of growing out the side of a tree, but this is actually caused by a bacteria. So this is caused by a bacteria called Agrobacterium tumefaciens, or we just say agro for short. And so what agro does is it makes a small piece of DNA and it injects it into the plant cell. And once it's in there, because DNA is molecular information, it tells the cell to grow bigger and bigger and bigger, to make lots and lots of sugar, and to kind of grab those nutrients and those resources from other cells around it. And that's what you get in the end, is these big tumorous looking growths on the side of a tree. So what biologists have done is we have actually engineered agro to not necessarily inject that DNA that's going to do bad things for the plant, but we can actually use agro to inject DNA that we want into the plant. And so all we have to do now is get our DNA into agro, and then we can get it into our plant. And so first thing we need to do is get it into agro, and so we're back to the same problem. So how do you get DNA within the cell? And so for agro, it's actually pretty simple. So we're going to shock it. And so you mix some DNA, so the kind that we had in our tube over there, with some bacteria. And so what happens when you shock the bacteria is the little cell will open up really quick and then it'll close again. And so hopefully if our DNA is close enough, it will kind of sneak inside after we shock the bacteria. And then from there we can just grow it up, and then we can inject it into a plant, and then hopefully it will inject it into a cell and we can see our protein. So let's finish this process up real quick and show you guys how that works. Okay, so again, here we have our template that has our DNA, tells us how to make our protein, and attached is the DNA that says how to make one of those fluorescent molecules. So we're going to mix this with some bacteria, and then we're going to shock it. So first, we need some of our DNA. We need a tip. Okay. Got some of our template. And in here is a whole bunch of bacteria. I can't tell you exactly how many because they're so teeny tiny and small, but there's quite a bit. So I'm going to mix our DNA that we just manipulated with the bacteria and mix it up a bit. Cool. Alright, so now we're going to take this bacteria and we need to put it into something that we could use to shock it. Okay. So this is a cuvette, so you can see the two little metal things on both sides. So we can put a charge through here and that'll shock all the cells really quick. So I'm going to add this inside. Make sure it's nice and even. Alright, sorry little guys. So again, this machine will shock our bacteria, and if our DNA is on the outside of their cells, it may sneak its way in. That little noise means it worked. Cool. Alright, So from here, all we have to do is grow the bacteria up, make sure that it has the DNA that we want inside, and then from there, like I said before, we can infect the plant, and hopefully it'll inject some of the DNA that we're interested in into our plant, and that'll tell the plant to make our protein with that special molecule attached. So let's infect some plants. I'm going to grab a syringe real quick. Alrighty. So this is a plant called Nicotiana benthamiana, or benthy for short. And we use this because these nice big juicy leaves are very easy to infect, and aggro likes going in there and infecting a bunch of cells. So here is our bacteria mix, and you guys may not be able to see, but there's tons and tons and tons of little bacteria in here. And so I'm going to take this and I'm going to inject it into a couple leaves right here. So draw out some of our liquid. Cool. Alright, now we infect. So, can you guys see that? A little bit. Alright. So you see that little liquid fill throughout the rest of the leaf, and so you're basically filling little pockets of air that are inside of the leaf, because plants need carbon dioxide to do photosynthesis. So we're replacing that air with this liquid, and that liquid has the bacteria in it. So I'm going to fill this whole thing. Maybe do this one just for fun. Oh, that's a good one. One fell swoop. It's cool. So now you have to give the bacteria some time to kind of grow and to give it some time to infect some of those cells. And so this is going to take a couple days, but I have an image of one that we took, or actually we took, someone else in another lab took on the computer over there, and I'll show you guys what it looks like when you look underneath a UV light. Because again, if our molecule that fluoresces is attached to our protein, we can see it. Cool. So again, this is a benthy plant, or Nicotiana benthamiana, and you can see that benthy kind of glows that pinkish color. And so there are some pigments or molecules within the plant that will fluoresce or glow in that nice red color. But if the molecule that we attach grows yellow, which it should, we can see our protein. And if you see that leaf right there that we infected, there's a bunch of teeny tiny little yellow dots on there. So maybe that's our protein, maybe our experiment works. So let's check it out underneath the microscope. And so I took these images a couple days ago. So this is an image of a cell when you just shine regular light on it. So you see this kind of like little puzzle piece looking shape outline on here. And then here's what happens when you shine UV light on this cell. Oh, so you guys see kind of that outline there. So if our DNA that we put in there tells the cell how to make our protein, and attached to that is a molecule that fluoresces, we should be able to tell where in the cell our protein is. And so here's what it looks like when you overlay the two images.